Viruses have been named through the years in a number of different ways, which is a little bit unfortunate because there is no sort of unifying system for naming viruses. Some viruses are named, for example, by the disease that they cause. The measles virus, the influenza virus, the rabies virus. Other viruses have been named by the geographic area where they were found. For example, there's a virus called Ebola Zaire. Some viruses have been given the name of the researcher who discovered them, like the Epstein-Barr virus. Some viruses are named by their particular shape or morphology. The coronavirus is an example of that. Researchers who saw the coronavirus under the microscope thought it looked a little bit like a crown, thought that the spike proteins that surrounded that virus made it look like a crown, and that word corona translates as crown. And of course, there are some viruses that are named using a combination of all these things, and a good example of that is the pandemic SARS-CoV-2 virus. Severe acute respiratory syndrome, the disease it causes. Coronavirus, crown shape, number 2. I want to talk just a minute about the influenza viruses and the difficulty of naming. This is probably the most complex naming system that you'll run into, the system of naming influenza viruses. There are 4 types of influenza virus, 4 very broad types called A, B, C, and D. These letters actually refer to the fact that among influenza viruses, there are 4 different categories of antigens in the virus. Now, if you're not familiar with that word antigen, an antigen is simply a molecule that is capable of eliciting an immune response. So there are 4 different antigens in the 4 different types of influenza viruses. Now, type A and type B are found in a variety of different animals, including humans. So yes, humans are not the only animals that can get influenza. Types A and B are the types of influenza that cause what we refer to as seasonal flu, the kinds of seasonal outbreaks that you see. Type C influenza is capable of causing disease in humans, but it causes a very mild disease. You wouldn't realize you had influenza with a type C. And type D doesn't even cause disease. In humans, it only affects cattle. So we won't even continue to talk about C or D. The influenza A viruses will be the focus of what I talk about with regards to naming them, because it's the influenza A viruses that are capable of causing the kinds of pandemics that we've seen in history. Now, if you look into that type, into that A type of influenza, there's a bunch of subtypes. Very, very similar to what we think of when we talk about strains of bacteria. There are different subtypes of influenza A, and they're named based on 2 of the spike proteins that you can see on the surface of the virus. There's an H type of spike protein and an N type. That H stands for something. It stands for hemagglutinin. That's the name of the protein. And the N stands for a protein called neuraminidase. There are actually 18 different types of the hemagglutinin protein. And there are 11 different types of the neuraminidase protein. Okay? Hence the numbering system. You've probably heard of H1N1. When you hear that there's an influenza virus named H1N1, it means it has the H1, the hemagglutinin type 1 spike, and it also has the neuraminidase type 1 spike. Yikes! So many numbers. Within the H1N1 subtypes, there are also strains, so sort of subtypes. It gets very complicated when we get into naming these viruses. I want to point out that the influenza B type, which does cause disease in humans, that can cause serious disease in humans, it doesn't have that H1N1 naming or the H number, N number subtype naming, but you will read about different strains of influenza B. Back in 1980, because there were so many influenza viruses and we needed a sort of a universal way to give each virus a name, the WHO, the World Health Organization, came up with a naming system, and it is complex. So, when you read the actual scientific name of any particular influenza virus, it's going to include the antigenic type, so whether it's A, B, C, or D, although no D's because that one infects cattle. It's going to include the host of origin. So where did this influenza virus come out of? So these might be things like pigs or swine, equine, which means horses, avian, chickens, ducks, so on. If the virus originated in humans, no host of origin is listed. So if there's no host of origin in the name, you know it came out of humans. It originated in humans. The next part of the name is the geographic area where it emerged. So this is really either the name of a city or the name of a country. Part 4 is the different strain of the influenza virus, and this is typically a number. Then they add in the year that this virus was first isolated, and if it's an influenza A, they include the H and N designation. So, every year, there are new influenza viruses for us to guard against, because these viruses, again, can cause very significant disease. They can mutate, and they do mutate often. So we're faced with new versions of influenza, with new traits that can become epidemic in places and even create pandemics, where the disease outbreak affects the entire globe. So luckily, we have developed influenza vaccines to at least help protect against the death, the disease, and the death that comes with influenza virus infection. So each year, we produce a new influenza vaccine. Some of these are what we call trivalent, and sometimes they are quadrivalent. Those terms just mean either they have 3 strains of the virus in them, that's trivalent, or 4 strains, that's quadrivalent. Now you might say, how do they know which strains to put in the vaccine? Well, it comes from public health systems that do surveillance, that watch around the globe for new strains of influenza that emerge. And lucky for us, I guess, Asia goes through their flu season before we go through ours. So by watching what happens in Asia every year, and in other parts of the world, we know which strains of virus are emerging and which ones are most likely to cause significant disease in the Western world. So, based on the prevailing strains, and also based on a subcommittee of the FDA a group of officials at the FDA who sit down, look at the data, and make the choices for the trivalent and the quadrivalent vaccines. So take a look at the 2019-2020 trivalent influenza vaccine. Look at the three viruses, the three strains of influenza that went into that vaccine. This is what I mean when I say the naming of influenza viruses is very complex. The first strain was A. Brisbane 02 2018 H1N1 PDM09-like virus. Remember, you give the strain number, you give the geographic location. You would tuck in here if it emerged out of a particular animal species. This one doesn't have a host of origin name, so you know it emerged out of humans. It originated in Brisbane. It's strain number 2. It emerged in 2018. It's the H1N1 type, and it has a strain number, PDM09. Very, very complex. The second one was A. Kansas, emerged in 2017, again from a human host of origin. That was an H3N2 type. The third strain was an influenza B type that emerged out of Colorado in 2017 and was the B/Victoria strain. Very complex naming system for influenza. But again, it's because there are so many individual strains of influenza, and there are new ones emerging all the time because of mutation. Now, outside of the naming problem with viruses, there is a system of classification for viruses that we need to be familiar with. It's called the Baltimore system. So this is the one system that's out there that helps us organize viruses, classify viruses. And it does that according to 4 things. First, the genome type of the virus. So whether it uses DNA or RNA, and whether that molecule is single-stranded or double-stranded. Next on the list is the sense designation. Now, I don't want to get into this too much, but this is only a feature of single-stranded genomes. So this sense designation only applies to the single-stranded RNA materials, and it's a system that names them either plus or minus according to the sequence of bases that you find in them. And again, I'm not going to get into too much detail on that, but that's called sense designation. The third characteristic that is considered in the Baltimore system is whether or not the virus has something in it, a molecule in it called reverse transcriptase. We'll talk about that in just a minute. And finally, the mode of replication of the viral genetic material. In other words, what process the host cell uses to copy the viral genetic material. So these 4 things are considered for each virus. Now I put this picture in because just in case you ever make the mistake of thinking about viruses as being very simple things, this picture, I think, drives home that they are not simple at all. On the left-hand side, this is the Baltimore system broken down into the 7 categories that viruses can fall under. I know it's hard to see, but they're showing us, again, these 4 characteristics. Particularly highlighted is what the genetic material looks like, whether it's DNA or RNA, whether it's single-stranded or double-stranded, what its sense designation is, whether it has reverse transcriptase in it, so on. All of these are different categories that viruses can fall under. Over here, in this little box, is all of the rest of us. all of the living cells on planet Earth, because of course we all use double-stranded DNA with the same sense designation. None of us have reverse transcriptase inside of us, and we all use the same mode of replication for our DNA. So here's us, here's living things, and here are all of the different options for viral particles. Let's leave viral naming and viral classification now and start talking about the hosts, the host organisms, the host cells that become hijacked with viruses. The first thing to know is that all cells on planet Earth, all types of cells, all cellular life can be infected by viruses. So all three domains, in other words, can be infected by viruses. Bacteria can be infected by viruses, archaea, and eukarya. So there's no living organism on this planet that is safe from viral infection. Viruses are host specific. Now, what do I mean by that? Any one particular virus has a particular host or a short list of hosts. So they can't just infect everything. The other thing to know is that if a virus is specific for a particular host, that specificity can change. And it changes over time. And it changes because of mutations. In order for a virus to be replicated, in order for more of it to be made, it has to get in to a host cell. And what that means is there has to be an attachment receptor on the surface of the membrane of the host cell. The protein on the surface of the virus, whether it's the spike protein or some other protein, the protein on the surface of the virus has to be able to bind to a protein on the surface of the host cell. That is what allows a viral particle to get into a cell. Now, sometimes the viral particle will only match with one kind of receptor protein on one cell type. Influenza is a good example of that. The kinds of cell receptors that an influenza virus can bind to are only found on one kind of cell. And that one kind of cell is the epithelium that you find in the respiratory tract. It's different for other viruses, though. Some viruses can bind to a receptor that is found on lots of different types of cells. In other words, on the surface of cells, sometimes there are protein receptors that are found on many different types of cells. The Epstein-Barr virus is like that. The receptor that Epstein-Barr binds to can be found on multiple different types of cells in the human body, which is why the disease that Epstein-Barr virus causes is so complex. It involves several different organ systems. Another example is the SARS-CoV-2 virus, this coronavirus. Coronavirus binds to a receptor called the ACE2 receptor. Now that stands for something. It's angiotensin converting enzyme 2. Now, you don't need to understand the details of that receptor, but it happens to be involved in the regulation of blood pressure. And that receptor happens to be found all over the human body. It's found in the lungs, yes. It's found on the surface of the epithelial cells that line the alveoli, those little grape-like sacs that are found in the depths of your lungs. The ACE2 receptor is found way down there inside your lungs. It is also found on the surface of the cells that line your blood vessels. And remember, those are called endothelial cells. It's found on the surface of smooth muscle cells in the arteries. It's found on the cells that line the small intestine. The ACE2 receptor is found all over the place in the human body, which is why this virus, yes, it causes a terrible pneumonia in the lungs, but we're also discovering that it can cause other disease syndromes. It can cause cardiovascular problems. It can cause intestinal problems. We're still learning all of the different disease syndromes that SARS-CoV-2 is capable of causing. This is an image, a colorized image on the left-hand side of the slide, taken under the electron microscope of this troublesome SARS-CoV-2 virus. You can see the spike proteins, the corona, the crown, surrounding these individual particles. Over here on the right, this is an artist's depiction of the SARS-CoV-2 virus, again with its spike proteins. The yellow, or this cream-colored area, represents the outside of a cell in the human body, and the blue area represents the cytoplasm inside of the cell. And this light color, light blue, represents the membrane. So they've depicted the ACE2 receptor sitting in the membrane. It's this green structure right here. And they've shown us that this pink spike protein can fit into this green receptor. It's sometimes described as a key fitting into a lock. I've heard the analogy of a baseball fitting into a glove. Those two things have to match each other. The protein in that pink spike has to be the correct shape to fit inside the protein in this green receptor. If those two things fit together, this happens. The virus is allowed to come into the cell. Now, I said that host virus specificity can actually change over time. And the reason that a virus changes hosts is because of genetic mutation. Genetic mutation is actually, relatively speaking, an uncommon event. It doesn't happen every second of every day. It's a relatively uncommon event. And that explains why, for example, we don't have pandemics every year. We have had a number of them, but we don't have them very frequently. Mutation is uncommon, relatively speaking. It's also random. And what that means is that sometimes mutations occur in viral particles, but it doesn't mean that the virus changes hosts. Now, some viral particles tend to mutate a lot. I already mentioned influenza. Luckily for us, so far at least, the data suggests that the SARS-CoV-2 virus seems to be mutating slowly. That's actually good news for us as the host of that virus. Mutations, which again, that word means a change in the order of the bases in the genetic material. Mutations change traits. And when a virus gets a new trait, sometimes it impacts the host, because it means sometimes the virus can attach to brand new cell types. Sometimes it can attach to brand new hosts. It depends. It varies. Now I've got a name of a virus listed here, a very long one. SARS lowercase r, RA, bat, CoV, RA, TG13. Yes, this is the name of a virus. This is the coronavirus that infects bats, that is believed to have been the source of the SARS-CoV-2 virus. So in other words, this is the virus that used to exist that infected bats, that underwent mutations to become SARS-CoV-2. When scientists compared the bases in the genetic material of the bat virus side by side with the bases in the SARS-CoV-2 virus, 96% of the bases matched between those two viruses. So this is our current best guess for where SARS-CoV-2 came from. It was existing in bats, infecting bats. It underwent some mutation and it changed hosts. And now it can infect humans. So mutations give viruses new traits. And again, sometimes it means new hosts. Sometimes it means new cell types in the old host. Sometimes it means that they can attach to receptors better. Sometimes, in other words, viruses that could bind to a particular receptor undergo a mutation that makes them bind to that receptor much more easily. So they can get into cells much more easily. They're more pathogenic, in other words. Right now, there's a lot of work in surveillance going on around a type of influenza called avian influenza. This is an influenza virus that infects birds, like the name suggests. And this is a particularly bad influenza in that it's quite pathogenic. It causes disease quite easily. Now, we've been watching this virus in Asia. We've been sampling it, and we've been sequencing the bases and watching for mutations. And we know that it's been mutating because influenza mutates a lot. What we are afraid of regarding avian influenza is that it will jump. It will jump hosts. It will mutate to the point where it can not only infect birds, but it can also infect humans. What we have discovered so far is that the mutations that the virus has gained has allowed it to infect humans in certain cases. In other words, there have been cases in Asia where people who have been exposed to sick birds have caught the virus from the bird. That's very scary, I know. There have been a handful of those cases reported. Luckily for us, there has been no reported transmission from human to human. In other words, this avian influenza virus has mutated and gained the new trait of being able to be transmitted from a bird to a human. But it doesn't have the trait it needs to go from human to human. Now, it may gain that trait at some point, and if it does, we want to be ready so that we can respond and try to prevent, again, another pandemic if possible. So, viruses have to bind to receptors on cells in order to get into the cell. That's the first step of viral infection. What happens exactly when a protein on a virus binds to a protein on a host cell is that those two proteins that have come together and bound to each other, they change shape. And it's that shape change that allows entry into the host cell. Sometimes the viral particle just injects its genetic material. Sometimes the entire viral particle goes into the cell. It varies. But once that material is in the host cell doesn't know that it came from a virus. The host cell can't tell the difference, in other words. When a host cell sees a piece of genetic material, it's just a piece of genetic material. So the host cell enzymes, the host cell machinery, is going to do with the viral genetic material what it does with its own genetic material. It's going to make copies of it, number one, and it's also going to transcribe and translate the genetic material. Remember from the central dogma, the process of taking DNA and making RNA, that's called transcription. And the process of taking RNA and making protein, that's called translation. So not only will the host cell make copies of the viral genetic material, it will also use it to make viral protein. The host cell is going to do all the work, in other words, for this parasite, this viral particle. And once all those things have been done, the viral proteins will assemble themselves into new viral particles with the newly copied viral genetic material inside. And then those newly made viral particles will burst out of that host cell and go on to infect other cells.